The overall goal of this procedure is to harvest and quantify fluorescently tractable bacteria from an intermediary host for subsequent infection of mammalian host cells. Here, Legionella pneumophila expressing GFP are used to infect the natural host protozoan Acanthamoeba castellanii monolayers. The L. pneumophila are then harvested by selective lysis, and the concentration of bacteria is then calculated using a formula based on fluorescence emission. Dilutions of protozoan-primed bacteria are then used to infect mammalian cell monolayers. Finally, the number of infectious bacteria in the mammalian cells are determined using a combination of fluorescence microscopy, flow cytometry, and plating lysates to recover colony-forming units. The resulting data demonstrate a pathogenic advantage gained for the bacteria that were cultivated in protozoan cells in the priming stage when compared to the same strain that was cultivated in artificial medium.
We developed this technique by observing that the expression of particular virulence factors in bacteria could not be induced through in vitro cultivation alone. Typical infection models of cultured monolayers of cells use in vitro cultured bacteria. For Legionella and several other bacteria, protozoans provide a natural reservoir for proliferation where the bacteria gains a pathogenic advantage. The technique that we will describe allows for quantification of protozoan-primed bacteria in order to accurately measure the contribution of a virulence factor that is only produced in the context of intracellular growth. Demonstrating the procedure will be Ben Doron, a research assistant, Drennan.